Imagine being invited to a government experiment where the scientist says, "We'll serve you 3 delicious meals every day and we'll just sprinkle in some chemicals that might kill you." That was real, and it sparked public pressure that led to the 1906 Pure Food and Drug Act, the first step toward food protections we now take for granted. In 2025, AI is accelerating drug discovery by predicting molecular properties and narrowing down candidates. Lab-grown meat has received limited approvals and is moving towards small-scale sale in places like the U.S. and Singapore. Today's tools reduce the need for risky human-first experiments, but they don't eliminate the need for careful ethics and testing. And to see how all of this began, we need to look at an era when almost anything could end up in your dinner plate. The Poison Squad, dining with danger. At the dawn of the 20th century, U.S. consumers had no guarantee of knowing what was in their food. Producers weren't required to label additives or prove they were safe. A family might buy milk only to discover it contained formaldehyde, better known as the chemical used to preserve corpses. Cuts of beef could be artificially made to look fresh with borax, a laundry additive. Even fruits and vegetables could be brightened with doses of copper sulfate, a toxic compound that lent a deceptive green sheen. These ingredients weren't exceptions, they were routine. To most shoppers, they seemed normal, just as we accept preservatives on ingredient labels today. But doctors were beginning to note unsettling links between unexplained illnesses and these hidden additives. That growing concern set the stage for one of the strangest government experiments in American history, the formation of the Poison Squad. Wiley's Poison Squad began in 1902 and involved about a dozen healthy young male volunteers. The plan reflected a simple idea, though it now sounds more like a grim provocation than a scientific study. If no one knew how food chemicals affected the human body, why not test them directly on people willing to eat them every day? Harvey Wiley, a government chemist, believed that only hard evidence could drive lawmakers to change. He argued that showing real human consequences would be more persuasive than any amount of rhetoric. With a small government budget, he hired a chef, set up a kitchen in Washington, D.C., and promised free meals to the recruits who agreed to have their diets laced with substances like borax, formaldehyde, copper sulfate, and sodium benzoate. On the surface, the deal almost sounded like a privilege for young men of the capital. 3 meals a day cooked professionally and served on schedule. For students or clerks living on tight wages, it was tempting. The hidden condition was that any plate, bread, stew, or butter, might contain one of the suspect additives. A slice of bread could be baked with salicylic acid. Milk might carry a drop of formaldehyde. Roasted meat might be dosed with borax. The chef didn't announce which dish held the chemical. The point was not choice, but endurance. Once the squad committed, they became test subjects under constant observation. Each day, their weight was measured and their meals were documented. Physicians collected blood and urine and tracked their digestion. Volunteers kept records of symptoms, headaches, nausea, fatigue, stomach cramps. The project demanded more than curiosity. It required resilience. Within weeks, several men were experiencing severe pain, digestive problems, and exhaustion. Some spent days confined to bed. Yet pride and public attention kept many from quitting. Their perseverance was praised as patriotic sacrifice. To a modern observer, the entire arrangement highlights how casually science once brushed aside ethics. The volunteers knew they were consuming poisons, which not only exposed them to risk but also introduced bias into the trials. The group was small, entirely male, and hardly representative of the public. Control measures were weak. By today's standards, the design would fail nearly every test of credibility or safety. Yet despite these flaws, the study achieved its central purpose. It showed how harmful the additives could be. News of their suffering filtered into newspapers and became widely discussed. Readers encountered vivid descriptions of young men doubled over after formaldehyde-laced meals, or noting the unmistakable metallic aftertaste in butter infused with copper compounds. The spectacle was both disturbing and compelling. Public outrage grew. For the first time, citizens clearly saw that their daily bread and milk were products of industrial chemistry with no safety oversight. That outrage helped change national policy. Public reporting of these trials helped build momentum for the 1906 Pure Food and Drug Act, the cornerstone of federal food regulation. While the act did not immediately ban every unsafe additive, it forced companies to adopt standards and face penalties for selling unlabeled or deceptive products. Over time, it evolved into the framework that gave rise to the Food and Drug Administration. In hindsight, the Poison Squad illustrates the sharp divide between reckless methodology and meaningful reform. These men intentionally ingested chemicals now regarded as dangerous, enduring cramps, bouts of illness, and long-term damage, all to demonstrate that food required protection from tampering. Their ordeal created undeniable proof at a time when lawmakers demanded more than theory. The legacy is still visible in the fine print labels on modern packaging. Today's shoppers scan for allergens, artificial colorings, or calorie counts. Behind each detail lies a century of regulation that began with volunteers trading their health to expose everyday poisons. The idea of pouring embalming fluid into milk seems unthinkable now precisely because of the squad's ordeal. Regulations that we treat as routine disclosures began as safeguards against those hidden hazards. But the Poison Squad's headline-making drama wasn't the last time Americans would learn that secret ingredients could be deadly. Their test kitchen may have launched the first wave of reform, but it only addressed part of the larger problem. Within a generation, a different kind of chemical disaster would strike medicine instead of food, bringing tragic consequences on an even greater scale. When medicine met tragedy, in the late 1930s, American doctors were eager to prescribe new treatments for infections, and one product seemed especially convenient: Elixir sulfanilamide. The active ingredient, sulfanilamide, had already earned a good reputation as one of the earliest antibiotics capable of fighting bacterial illness. The danger came not from the drug itself, but from the way one company reformulated it. To produce a liquid version, the manufacturer mixed sulfanilamide with diethylene glycol, a compound later recognized as highly toxic when metabolized, leading to kidney failure, nervous system injury, and even blindness. Raspberry flavoring was added to mask the bitter taste, and the mixture was bottled and shipped nationwide. At the time, no federal law required safety testing of such formulations before they reached pharmacy shelves. Patients of all ages, trusting the prescription and the label, began dosing themselves with what was essentially poison disguised as medicine. Doctors quickly noticed something disastrous. Patients who had been recovering on conventional sulfanilamide tablets declined rapidly after switching to the syrup. Many experienced sharp abdominal pain, vomiting, and mental confusion, followed by kidney shutdown. In town after town, people who seemed on the brink of recovery from infections instead collapsed within days. What alarmed physicians most was how consistently and quickly the new liquid led to fatal outcomes. Healthy children who had only minor illnesses were among the first to die. In a single week, some communities lost several young patients, leaving doctors stunned at the sudden pattern of deaths. The scale of the disaster escalated within weeks. Hospitals saw waves of patients with the same symptoms, and morgues began to fill with victims who never should have died. Families who had gone to their doctor expecting safe treatment instead found themselves grieving unexpected loss. One account tells of parents spoon-feeding their daughter with confidence that the raspberry-flavored elixir was helping her infection, only to watch her grow weaker by the hour. Across the country, similar stories repeated, each one more devastating for having come from a drug that was legally sold. What made tragedy even harder to accept was that the company had not technically broken any law. Federal rules at that time only required that drugs be labeled honestly and not adulterated in ways that altered identity. The bottles of Elixir sulfanilamide did list their ingredients. What they lacked was proof of safety, a requirement that did not yet exist. Regulators had the ability to act if a medicine was deliberately misbranded or falsely labeled, but they could not demand toxicity studies before products reached the public. The deaths, in effect, were the result not of deception but of regulatory absence. As more fatalities came to light, ultimately at least 105 men, women, and children died. Federal agencies scrambled to contain the situation. The Food and Drug Administration organized one of the largest recalls in its history up to that point. Inspectors fanned out to track every case of the elixir, contacting doctors, visiting homes, and warning pharmacies. Radio broadcasts urged Americans to stop using the medicine. Despite the effort, the recall could not outrun the toll already underway. By the time the last bottles were collected, the damage was irreversible, and public confidence in the safety of medicine had been cracked open. For Congress, the lesson was undeniable. A system in which new products could be concocted with solvents or dyes and sold without safety checks was no longer acceptable. Outrage pressed legislators to act swiftly, and in 1938 the Federal Food, Drug, and Cosmetic Act became law. For the first time, pharmaceutical manufacturers had to demonstrate through studies that a product was safe before it could enter the marketplace. Regulators gained authority to inspect plants, demand data, and set standards beyond simple misbranding. By mandating pre-market approval, the law filled the gap that had allowed such a catastrophic release in the first place. The transformation of the pharmaceutical industry after 1938 cannot be overstated. Entire procedures of production and testing changed overnight. Companies were required to think about toxicity, dosage, and long-term effects in ways they had previously ignored. What had been a field focused on innovation and speed now had to account for public health safeguards as a condition of doing business. The tragedy of Elixir sulfanilamide cemented the principle that safety is not optional, it is integral to the legitimacy of medicine. This reform carried invisible but profound benefits a generation later. When thalidomide emerged in the late 1950s as a treatment for morning sickness, it spread rapidly across Europe, leading to thousands of infants born with severe deformities. In the United States, however, thalidomide never gained wide approval. The difference was the regulatory power granted by the 1938 Act. FDA scientists, relying on their authority to demand data on developmental safety, refused to clear the drug without more proof. That caution spared countless American families from the suffering experienced abroad. The barrier that stood between tragedy and public health was the law born out of the sulfanilamide disaster. The ripple effects extended beyond one near miss. Over time, the expectations established by the 1938 Act formalized into structured clinical trials, phase-based testing, and long-term toxicology studies. Research moved from scattered experiments to systematic evaluations that included animal studies, dose ranging, and later, structured human trials. Each step was designed to prevent a repeat of the mid-1930s catastrophe. Even industry began to recognize that public trust depended on compliance with these new standards. Far from resisting, many manufacturers learned that credibility in the market was inseparable from demonstrating safety. For ordinary Americans, the difference reshaped daily life. The simple act of picking up a prescription now carried a kind of assurance that hadn't existed just a year before. Patients came to expect that anything on a pharmacy shelf had been reviewed, tested, and cleared by experts. This shift in expectation restored trust. Physicians prescribing medicine could speak with more certainty that treatments would heal rather than harm, and families began to rely on a system that promised oversight. It is hard now to imagine a pharmacy without that safety net. Yet in the 1930s, this tragedy had to occur before the rules were written. While the loss was devastating for the families involved, its consequence was the creation of protections that became foundational to all future regulation. The Elixir sulfanilamide case remains a somber reminder that medical progress cannot depend on trial and error at the cost of human lives. Instead, it must build on structured safeguards that serve the public. By the end of that decade, Americans had entered a new regulatory era in medicine, one built not just on optimism about new compounds, but also on guarantees that those compounds would not kill the very people they aimed to treat. Yet even as the nation adjusted to these stricter rules, other discoveries were arriving from unexpected corners of science. Some came not from medical laboratories at all, but from industries with little connection to health, where chance experiments would open the door to an entirely new arsenal against disease. Science by accident, antibiotics from red dye. One of the most surprising steps forward in medicine came not from a hospital or research laboratory, but from the dye industry. In the early 1930s, German chemists were developing synthetic colors for textiles when they stumbled across an acid dye compound known later as Prontosil. Unlike other dyes, Prontosil did more than stain fabric. When tested in animals, it showed clear antibacterial activity, helping them survive infections that were otherwise lethal. At a time when even routine illnesses posed serious threats, the idea that a synthetic red dye could protect against bacteria challenged every assumption medicine held about infectious disease. Prontosil itself looked unremarkable beyond its scarlet hue, but its testing produced results that were impossible to ignore. Researchers observed that animals given Prontosil during bacterial exposure survived in much higher numbers than untreated controls. The conclusion was startling. A man-made chemical designed for cloth could, under the right conditions, alter the course of infection. This was years before the rigor of randomized clinical trials, so persuading others would not be easy, but the evidence pointed to something profound. Skepticism was the immediate response. Doctors were accustomed to treatments that relieved symptoms, not remedies that struck at the cause. The suggestion that a textile dye might act as a medicine was almost laughable. Many dismissed the findings outright, labeling them as coincidences. Yet bits of clinical experimentation crept through in despite of doubt. Driven by desperation, some physicians tried Prontosil in patients suffering severe infections. Instead of decline, they saw recovery. Children with life-threatening streptococcal illnesses pulled through, and patients on the edge of death from blood poisoning stood a chance they would not otherwise have had. The turning point came with chemical analysis of the dye's journey through the body. Researchers discovered that Prontosil was metabolized into Sulfanilamide, a smaller, colorless molecule that was actually responsible for the antibacterial effect. The dye acted as a carrier. The metabolite did the real work by blocking bacterial growth. This insight bridged the gap between strange observation and scientific explanation. Understanding the mechanism opened the door to a new class of therapies, sulfa drugs, setting off a wave of discovery in medicinal chemistry. Once sulfanilamide was identified, its potential spread quickly. Laboratories modified its structure to create derivatives, each with slightly different properties and applications. Soon dozens of sulfa drugs were in circulation, offering tools against infections that had previously killed indiscriminately. This was the beginning of the antibiotic era, and its effects touched nearly every aspect of daily life. Pneumonia, which had been a common cause of death, could now be treated with consistent success. Infections after childbirth became manageable. Surgical operations lost much of their risk of fatal infection. Everyday medicine had crossed into a new stage, and it happened because a dye company linked color chemistry to bacterial survival. The practical impact was felt most during the Second World War. Historically, more soldiers had died from infections of their wounds than from combat itself. Sulfa drugs changed that equation. Medics could administer tablets on the battlefield, reducing the threat of blood poisoning and saving countless lives. Civilian populations also benefited. Families saw children rebound from illnesses that had once been funeral sentences. For the first time in human history, chemical intervention could offer a consistent defense against bacterial disease. The chemist responsible for this line of work was later awarded a Nobel Prize in recognition of how transformative the discovery had been. Not every physician embraced the drugs right away. Resistance to industrial chemicals as medicine remained strong, and old remedies still lingered in practice. Adoption required repeated success stories, as well as social pressure from patients who demanded the new treatments they saw working for their neighbors. Eventually, regulations and national health agencies recognized sulfa drugs officially, removing their fringe reputation and embedding them in mainstream pharmacology. However, their record carried a warning sign that stretched into the future. Bacterial resistance began to surface. Infections that once responded strongly to sulfa drugs sometimes returned more difficult to treat. The lesson was unmistakable. The effectiveness of antibiotics would never be permanent. Microbes changed, adapted, and pushed back. Even as penicillin and other families of antibiotics emerged, the fragility first revealed by the sulfa era stayed in view as a constant medical challenge. Prontosil and its derivatives remain remembered not only for their immediate impact, but also for how they reshaped the very framework of drug development. The story illustrated the value of unexpected discovery, the need for patience in adoption, and the realities of biological limits. What began a textile laboratory forced medicine to recognize that life-saving chemicals might emerge from the most unexpected of places. By the midpoint of the 20th century, antibiotics were deeply embedded in both civilian and military medicine. Yet their story also showed how science could swing from unintentional breakthroughs to devastating oversights. Success bred optimism, but optimism could obscure risk. The next major test of medicine's relationship with chemistry came not from an accidental dye, but from a drug that promised relief in everyday life and instead delivered tragedy on a global scale. The Thalidomide lesson, chemistry's cruel twist. In 1957, a new medication appeared that seemed to answer a persistent challenge of pregnancy, morning sickness. The drug was thalidomide. Advertisements promoted it as safe, non-addictive, and effective not only against nausea, but also as a sleep aid and sedative. Physicians prescribed it widely, encouraged by claims that it avoided the risk of earlier tranquilizers. Expectant mothers embraced what many saw as a modern pharmaceutical convenience, and by the early 1960s, thalidomide was distributed across Europe, Australia, and other regions in millions of doses. But within just a few years, hospitals began reporting a surge of unusual and devastating birth defects. Infants were born with severely shortened limbs, sometimes resembling flippers, or with missing ears, malformed facial features, or internal abnormalities that made survival impossible. By 1961, it was clear that a common thread linked these tragedies: mothers who had been prescribed thalidomide in early pregnancy. Thousands of infants were affected, and families faced heartbreak not from rare genetic circumstances, but from a drug sold over the counter in brightly printed boxes. What was intended as a source of relief became a catastrophe visible across continents. The grief was compounded by betrayal. Women had reason to believe they were making a safe choice on the guidance of doctors and the assurances of companies. Instead, many carried guilt when their children were born disabled or did not survive infancy. The psychological toll stretched beyond mothers. Fathers, entire families, and healthcare providers confronted a reality in which an approved medicine had inflicted unimaginable harm. The sense of trust between patients, doctors, and the drug industry fractured overnight. The scientific question of what went wrong pointed directly to chemistry. Thalidomide exists as two chiral enantiomers, molecular mirror images compared to left and right hands. One produced a calming and anti-nausea effect that had made the drug appealing. The other profoundly interfered with fetal development, disrupting growth of limbs and tissues. Regulators at the time had not required close study of stereochemistry in drug candidates, assuming a single formula represented a single effect. But thalidomide shattered that assumption. Even worse, the enantiomers could interconvert inside the human body. This meant that even if manufacturers had marketed only the safer form, once ingested it would convert into both versions. In vivo racemization ensured there could be no truly safe enantiomer of thalidomide for pregnant women. What had seemed like a technical detail in molecular geometry became the deciding factor between healthy development and catastrophic birth defects. The catastrophe unfolded just a generation after the United States had put the 1938 Food, Drug, and Cosmetic Act into place in response to the sulfanilamide disaster. Those stricter standards required manufacturers to provide safety testing data before new drugs reached the public. That regulatory caution, coupled with FDA scientists reviewing thalidomide, slowed its approval in the U.S. and ultimately prevented the drug from being widely distributed there. The limited licenses granted elsewhere spared the United States the full extent of devastation seen in Europe, an outcome that revealed how the lessons of one crisis had prevented another from becoming worse. For countries where thalidomide had flooded the market, however, reforms became urgent. Clinical testing had to become more rigorous, particularly in pregnancy. Screening for teratogenic effects was no longer a minor detail but a core element of toxicology. Rules for informed consent tightened. Developers could no longer assume that effective relief for symptoms outweighed unknowns in vulnerable groups. Regulatory agencies across the world began demanding proof not only of general safety, but also of specific risk to fetal development, dismantling the old assumption that animal testing alone was sufficient insurance. The public response deepened these changes. Families raising children with disabilities brought new urgency to debates about oversight. Thalidomide became not just a chemical lesson, but a cultural one. Survivors visible in communities made the crisis harder to forget, reminding everyone that medicines carried consequences beyond marketing claims. The relationship between citizens and regulators grew less trusting, with greater public demand for transparency in drug approval and ongoing monitoring of risks. While the drug had earned permanent stigma, its chemical effects could not be erased. Later research showed that thalidomide could be effective for certain cancers such as multiple myeloma and for complications of leprosy. Doctors reintroduced it, but under strictly controlled conditions and with absolute prohibitions for use during pregnancy. In those narrow contexts, the drug proved valuable again, though forever shadowed by its past. Its presence on hospital formularies came with fortified safeguards, layered warnings, and stringent distribution systems. The legacy extended into scientific education. The story of thalidomide transformed stereochemistry from an abstract subject into a deadly serious lesson. Today, chemistry students are taught that two molecules can share the same atomic components, yet produce entirely different biological actions. The drug's history provides a concrete reminder that overlooking enantiomeric effects can produce devastation. The case established chiral analysis as a baseline requirement in every new drug's development pipeline. Ethical questions also sharpened. How should risk be balanced against relief, and who should decide when information is uncertain? Thalidomide had been marketed primarily for discomforts like nausea and sleeplessness during pregnancy, conditions unpleasant but not life-threatening. Against those modest benefits, the hidden risk proved catastrophic. Since then, regulatory systems have aimed to prevent similar trade-offs from passing unchecked. Informed consent gained weight, and the bar for prescribing to pregnant women was set far higher. The survivors themselves ensured memory would not fade. Many children born in the early 1960s grew to adulthood with disabilities, finding ways to live full lives but also advocating for recognition of what their families endured. Their visibility kept the tragedy part of public discourse long after the drug disappeared from general circulation. Their voices reminded society that behind regulatory laws and chemical formulas were real lives forever changed. Today's drug discovery frameworks reflect the scars of that period. From preclinical testing to computational modeling, the failure of thalidomide pushed scientists to anticipate risk at levels never imagined before it. Molecules are screened against pregnancy outcomes in lab animals, and data on stereoisomers is treated as indispensable. Advancements in three-dimensional modeling of molecular binding and interaction are the scientific descendants of lessons carved from the crisis of the early 1960s. Yet despite progress, the paradox remains: the same chemical that once inflicted irreversible harm now saves lives in cancer wards. Each prescription is surrounded by warnings, black box labels, and risk mitigation systems, but the dual identity cannot be forgotten. Thalidomide continues to exist as both a tool of healing and as a symbol of preventable tragedy. The narrow line between benefit and catastrophe has become clearer precisely because this drug revealed how fragile it can be. This dual legacy is why thalidomide remains so prominent in pharmaceutical history. It demonstrates the stakes of molecular handedness and the fragility of human oversight. The story is not just about chemistry, but also about responsibility. Proof that even a detail as small as geometric orientation demands complete attention when human lives are at stake. That stark reminder shaped the regulatory framework we live with today, a system in which every pill is the product of intense scrutiny. By the early 1960s, governments, scientists, and the public had seen how easily trust could collapse and how badly damage spread when chemistry was underestimated. Out of this failure came stricter standards and sharper tools. What followed was an era of medicine shaped not just by new compounds, but by the technology that made deeper testing possible, opening doors to analytical powers that would transform science in unexpected ways. Supercharging science, the calculator that changed everything. By the middle of the 20th century, another type of revolution was stirring, not in chemistry this time, but in machines that promised to take over the burden of arithmetic. Long before home computers or smartphones, the calculator became the first glimpse of digital thinking embedded in everyday life. At a glance, it looked like a simple number box, but behind the scenes, it was the consumer product that created the demand, financing, and technical leaps that laid the foundation for the entire microchip era. The earliest electronic calculators were hardly recognizable compared to the pocket devices of the 1970s. Many were the size of a desk, built from racks of vacuum tubes and mechanical switches. They hummed, glowed, and gave off heat, all in the service of tasks that a slide rule could solve more quietly. These models cost as much as a family car, and maintenance was a headache, replacing a single burnt-out tube meant fishing through hundreds of glass cylinders. Companies and government labs bought them, but for most people they were exotic and completely impractical. The important shift, however, was no longer about convenience. It was about proof. These machines showed that arithmetic could be turned into electrical signals, ready to be shrunk and refined. That shrinking came with transistors. Once tubes were replaced by small, durable electronic switches, machines that filled furniture-sized cabinets could be reduced to desktop boxes. Through the 1960s, prototypes steadily became cheaper, cooler, and more reliable. The biggest leap, though, arrived with integrated circuits. By condensing entire networks of transistors onto a tiny silicon chip, engineers could turn calculators from costly showcases into affordable tools. This made him one of the first consumer products to popularize IC technology. Companies invested in chip production lines specifically to meet calculator demand, and the success of pocket models directly funded the infrastructure that soon powered early computers. Consumers were not just buying faster math, they were underwriting a new electronics industry. Every student who purchased a calculator in the 1970s contributed indirectly to the scaling of semiconductor factories. Mass market popularity drove costs down and encouraged design improvements. That hunger for smaller, more efficient chips spilled outward, setting the stage for personal computers, digital watches, and eventually mobile phones. Without the calculator boom, semiconductor miniaturization might have taken far longer to reach the prices that enabled digital technology to spread globally. Displays offered another critical innovation. The first calculators used clunky light bulbs or power-hungry LED readouts. But to make a truly pocket-sized, battery-powered device, manufacturers needed something far more efficient. The answer was the liquid crystal display, or LCD. Calculators became one of the very first mass-market devices to adopt LCDs, proving they could deliver clear numbers using only a trickle of power. That success established LCDs as the default choice for portable electronics. A straight line can be drawn from calculator screens to the displays on wristwatches, laptops, and eventually smartphones. By convincing buyers that LCDs were reliable, durable, and clear, calculators helped overcome the barrier between experimental display technology and mainstream use. For students, the change was immediate and visible. 2 decades earlier, math homework had meant grinding through logs and trigonometry with paper tables or slide rule. By the late 1970s, a pocket calculator could process those same functions seconds. Science class experiments, engineering calculations, and even basic budgeting shifted from longhand to instant results. Teachers worried that reliance on buttons would erode basic numeracy, sparking fierce debates about whether calculators should be banned in classrooms. Despite the resistance, low prices and obvious advantages made them unavoidable. Within a generation, they became as ordinary as notebooks or rulers. In business and research, the adoption reshaped professional practice. Bank clerks could process accounts in real time. Architects adjusted figures without convening teams of assistants. Scientists plotting data could spend their time interpreting results rather than carrying out columns of multiplication. The speed was transformative, but perhaps more importantly, it normalized the assumption that machines belonged in thought processes normally handled by people. This expectation that computation itself should be offloaded was one of the calculator's most enduring cultural contributions. By the 1980s, entire markets had sprung up around specialized calculators. Financial calculators solved tax schedules, engineering models handled trigonometric functions, and scientific calculators became standard for higher education. The race for features wasn't just about pleasing students. It compelled chip makers to refine microprocessors to balance speed, complexity, and battery life. Each generation of calculators stretched the boundaries of hardware while teaching consumers to demand more power in smaller packages. That appetite for powerful miniaturization flowed directly into personal computing. The factories producing calculator chips soon adapted those lines to make processors for microcomputers. In many cases, the same engineering teams that perfected calculator logic used those lessons to develop computer architecture. Laptops incorporated the dense transistor layouts that calculators had helped validate. And when phones later required processors small enough to fit comfortably in a pocket, designers built on the same problem-solving methods first honed inside calculator chips. Artificial intelligence running on billions of operations per second might seem like a distant cousin to the calculator, yet the connection is close. At their core, both rely on rapid, reliable processing of numbers. The calculator's mass production serves as the early trial run for integrating circuits and displays at an everyday scale. That legacy lies under the hood of today's AI systems, which depend on cheap chips and energy-efficient displays, all technologies first popularized. Because consumers wanted arithmetic small enough to carry in their hands. Critics once dismissed the calculator as a lazy crutch, but history has proven otherwise. Far from undermining ability, it familiarized entire generations with digital logic and with the expectation that machines could act as cognitive partners. By forcing industries to compress hardware, refine displays, and mass-produce chips, calculators marked one of the quietest but most influential turning points in the story of modern technology. Seen in hindsight, the sight of hulking two machines rolling into offices seems almost comic. Yet each absurdly oversized calculator made way for the pocket devices that millions would later depend on. And those pocket devices, in turn, helped to finance the infrastructure on which today's digital systems rest. A simple curiosity, how to automate basic sums, grew into a path that connected laboratories, classrooms, and eventually the chips inside every smartphone. The calculator, then, is more than an accessory or a shortcut. It was the consumer gateway to digital life, the device that made microprocessors and LCDs common in homes, and the stepping stone that carried computation from lab benches to breakfast tables. Its influence is still visible in the logic that governs machines today. And as history shows, even the most trusted tools can obscure blind spots, assumptions that seem solid but later prove mistaken. Mistaken identities, fighting the wrong enemy. Doctors once believed they fully understood the enemy behind influenza, but for decades they were fighting the wrong target. Physicians in the late 19th and early 20th centuries treated flu as if it were a bacterial illness, a conclusion that fit neatly within the frameworks available at the time. Koch's postulates had given medicine a powerful guide. Find a microorganism in every sick case, grow it in a pure culture, introduce it into a healthy subject, and see the same disease appear. This method had solved elusive puzzles like tuberculosis, anthrax, and cholera. But the approach was not universal. Viruses could not be cultured independently like bacteria, and diseases with silent carriers or hidden stages, such as latent tuberculosis, already stretched the limits of Koch's system. Influenza, which spread rapidly and unpredictably, would prove an even more confounding challenge. The strongest candidate under the microscopes of that era was a bacterium christened Haemophilus influenza. It appeared often in respiratory samples from flu patients and to researchers eager for visible evidence that presence looked like causation. This assumption hardened into dogma. Public health authorities taught it, physicians accepted it, and entire prevention campaigns were built on it. In hindsight, the evidence was paper-thin. The bacterium was more an opportunistic passenger than the root cause. Still, it provided a visible figure to blame, something that could be stained, drawn in medical journals, and pointed to when patients demanded answers. When the 1918 influenza pandemic erupted with catastrophic force, that assumption was already codified. In the face of mass death, institutions pushed urgently to develop protective measures. What emerged were bacterial vaccines meant to shield populations against Haemophilus influenza and similar organisms. Entire communities lined up for inoculations, yet none of these preparations stopped influenza spread. The numbers told a stark story. People fell ill whether or not they had received the shots. Instead of preventing infection, these vaccines did little more than erode confidence in medicine. But instead of questioning the bacterial framework itself, researchers speculated about wrong strains, missing ingredients, or inadequate techniques. Few dared to challenge the foundation on which their science was built. The misunderstanding persisted because the alternatives were invisible. Bacteria could be cultured on plates and examined under a standard light microscope. Viruses, by contrast, lay far outside the resolution of those instruments, inferred only indirectly through filtration experiments. To many doctors, to blame a virus was to invoke a ghost. Governments wanted concrete microbes that could be photographed, cultured, and cataloged, and bacteria filled that role whether or not the evidence held. The needs of certainty, explanation, and policy outweighed the thinness of proof. The cost of this blind spot was immense. For decades, vaccination campaigns failed to reduce influenza outbreaks. Outbreak after outbreak challenged the bacterial model, but the inertia of established science slowed its abandonment. To admit error was to undermine not just a single theory, but the authority of an entire medical establishment that had relied on visual models of disease. Physicians who had built reputations on bacterial detection were hesitant to allow that a hidden agent beyond their tools' reach was the true culprit. The structure of knowledge, as much as the limits of the equipment, reinforced the error. It took a careful experiment in 1933 to undo the misconception. Researchers working with ferrets, chosen because their respiratory systems closely resembled those of humans, deliberately exposed them to material taken from influenza patients. The ferrets developed classic flu symptoms, even when the samples were filtered to exclude every detectable bacterium. The conclusion was unavoidable. The illness was caused by something smaller, something invisible to the microscopes of the day. In other words, it was a virus. The clarity of these results shattered the bacterial assumption that had dominated for decades. For the first time, the identity of influenza was accurately pinned to its true cause. The discovery was bitter and liberating at once. On one hand, it meant that almost half a century of medical effort had targeted the wrong enemy, costing time, resources, and most tragically, lives. The 1918 pandemic had unfolded under the shadow of this mistake, and millions died while physicians wielded vaccines that could never have worked. On the other hand, finally identifying influenza as viral freed science to adapt new methods, leading to animal models, serum typing, and eventually the development of effective flu vaccines. The mistake revealed the limitations of Koch's framework, while pointing the way toward more flexible, evidence-driven approaches. The episode left a lasting caution. It showed that reliance on one method, however successful in other contexts, could blind entire fields to alternative explanations. It reminded scientists that visibility is not the same as causation, that what appears in a cultured dish is not automatically the culprit of a disease. And it emphasized that humility, recognizing when gaps in knowledge exist, is as necessary to science as discovery itself. That memory informs modern practice. In 2025, when an outbreak emerges, researchers no longer assume a cause based on what looks simplest under a microscope. Genetic sequencing identifies pathogens at the molecular level, often within hours. Portable sequencing tools now operate in field clinics far from laboratories, transmitting data to global databases. Artificial intelligence compares genetic codes across continents, flagging anomalies and predicting which viral proteins may mutate next. Where generations once spent decades circling a false target, today's scientists can leap directly to verified results. But these advances rest not only on technology. They reflect a learned vigilance, the hard lesson that certainty must be earned, not assumed. Every time an AI platform identifies a novel sequence, every time epidemiologists test whether the suspected culprit truly reproduces illness, they carry forward the warning encoded in influenza's history. The mistake of misidentification was not just an episode to forget, it became a guidepost shaping the culture of biomedical science. It stands, too, as a sobering reminder of how much loss can accumulate when misdirection rules unchecked. For nearly half a century, humanity organized its defenses around bacteria that were never the cause. Families mourned, resources drained away, and epidemics surged without real opposition. Only with ferret experiments in 1933 was the error finally overturned, giving science the footing it needed to address the real agent. That mistake caused decades of misdirected research, so how did scientists finally change course? The answer came by shifting focus away from what could be seen to what could be traced. And once evidence finally pointed in the right direction, medicine had to learn to recognize patterns of spread, to track forces moving invisibly across crowded cities. Errors of causation gave way to questions of transmission, and that brought another defining chapter in the story of public health. The water pump mystery. Cholera's hidden path. London in the 1850s was a city swollen by the Industrial Revolution, where poverty and filth filled the narrow streets. Houses often lacked plumbing, sewage drained into cesspits or open gutters, and drinking water frequently came from streams and wells contaminated by human waste. In such conditions, disease moved quickly. Cholera was among the most feared, striking without warning and killing entire families in days. Outbreaks surged through neighborhoods suddenly, leaving devastation behind, while science struggled to explain why. Many officials blamed the air itself. The miasma theory held that foul odors, the stench of waste, rot, and decay, poisoned those who breathed them. To city leaders, this seemed logical. Entire districts reeked, and illness appeared to follow the stench. Reformers even focused more on cleaning up smells and actually removing waste, convinced that odor alone carried disease. One physician, John Snow, doubted this explanation. A doctor and anesthesiologist by trade, Snow had watched cholera return again and again, always with terrifying speed. To him, it made little sense that something as vague as bad air could explain the way certain households were devastated, while others only steps away escaped illness. He suspected a more direct pathway: water. In a city where sewage often flowed into the same rivers from which families collected drinking water, it was plausible that cholera moved not through lungs, but through what people drank. Snow's suspicion was not popular. Senior doctors dismissed the idea and clung firmly to miasma theory, which matched prevailing logic and experience. But during the outbreak of 1854 in Soho, London, Snow found an opportunity to test his hypothesis. The epidemic hit fast and hard. More than 500 residents died in just 10 days. Panic spread, families fled, and entire streets emptied. Snow took to the streets as well, not to flee but to collect evidence. He visited homes, recorded the number of cases, and then turned those records into marks on a map. Each death became a small black dot. As the dots gathered, a chilling pattern appeared. They radiated outward from a single pump at the corner of Broad Street. The correlation was too strong to ignore. Snow saw that households drawing water from the Broad Street pump suffered the brunt of the outbreak, while those with alternative sources were often spared. In contrast, a local brewery where workers drank mostly beer brewed from their own water supply showed almost no cholera cases, even though employees lived in the heart of the district. Brewing boiled water, destroying the invisible agent. This detail gave Snow another thread of proof. Cholera was not floating in the air, it was being consumed. Yet convincing decision-makers proved harder. Officials argued that people near the Broad Street pump might simply be inhaling more foul odors, making the pump a coincidence rather than the true source. But Snow pressed his case with relentless clarity, presenting his map and statistics to a parish committee. He showed, for example, that areas supplied with water downstream from sewage outlets had cholera rates up to 14 times higher than districts drawing from cleaner sections of the Thames. Numbers on paper spoke more forcefully than speculation. At last, the committee acted. They ordered the handle on the Broad Street pump removed, disabling its use. The local outbreak diminished, and many lives were almost certainly spared. Snow had proven that mapping disease and tracing patterns of exposure revealed truths that smell-based theories could not, without ever knowing the actual bacterium, Vibrio cholerae, which would not be identified until decades later. He had shown where and how cholera traveled. Snow's victory, however, was fragile. When the epidemic passed, many city officials drifted back to older explanations, still convinced foul air was a deadly force. Large sewer projects were delayed. Reforms focused on deodorizing neighborhoods instead of fixing contaminated water. And Snow's insights were relegated to the margins. But his map survived, shared among medical circles and studied by those who came after. Once germ theory gained global recognition late in the century, his work was recast as visionary. The map was no longer a curiosity, but a model of how to link human behavior, infrastructure, and disease spread. Snow's 1854 study became a foundation of modern epidemiology, introducing the idea that careful tracking, mapping, and statistical comparison could identify the source of outbreaks. His use of evidence over assumption set a new standard. Instead of vague theories of air and odor, he relied on tangible patterns: who got sick, where they lived, and what they consumed. That shift marked the beginning of outbreak investigation as a discipline. The echoes of his approach extend into the present. Where Snow drew his map by hand, public health teams now generate live dashboards that track case counts and geolocations in real time. In place of tallying deaths on the city blocks, officials analyze mobile phone location data, credit card transactions, and genomic sequences from patient samples. The principle, however, remains the same. Disease reveals its path if one carefully traces connections. Snow's Broad Street pump map is the ancestor of today's contact tracing and pandemic modeling systems. In 2025, as outbreaks unfold, AI systems do the work Snow once performed on foot. Algorithms sift through massive datasets to pinpoint likely sources and predict where clusters will appear next. Recommendations for lockdown zones or targeted aid reflect the same logic he used in London. Find a link, remove the exposure, and break the chain of transmission. Even smartphone apps that alert people to potential exposure carry the DNA of Snow's insight that shared resources and contact networks are the real scaffolds of disease. The Broad Street pump remains a symbol, not because of its iron handle, but because of the method it represents. Outbreaks cannot be solved with speculation about unseen vapors. They demand evidence, patterns, and data woven into coherent explanation. Snow's determination to gather facts paved the way for every modern health response, from tracing foodborne illnesses to predicting viral mutations. Reflecting on this history makes clear that science often advances not by discovering new tools, but by asking sharper questions with the tools already at hand. Snow walked through Soho with only paper, ink, and persistence, yet he anticipated the data-driven strategies that now shape global epidemic response. His map is as much a blueprint for modern public health as any digital algorithm. And as history shows, the heart of public health is often tested not in laboratories but in moments of urgent crisis when a community faces destruction unless swift action intervenes. In one such moment, far from London and decades later, a remote town confronted a deadly threat of its own, one where survival depended not on pumps or maps but on courage and an almost impossible delivery across ice and snow. Dogs, serum, and the frozen race against diphtheria. In the winter of 1925, the town of Nome, Alaska found itself staring down an outbreak that threatened to destroy it from within. Diphtheria, a bacterial infection notorious for coating the throat with a membrane that choked its victims, had begun spreading through the isolated settlement. Nome had a single physician, almost no supplies, and brutal geography cutting it off from outside aid. The only known therapy was antitoxin serum, hundreds of miles away in Anchorage. If it could not be delivered, the disease would claim entire families across Nome and nearby villages. Moving the serum across Alaska in January was a logistical nightmare. Sub-zero winds grounded planes, ships were frozen into port, and the railroad went only part of the distance. Nome might as well have been on another planet. Officials concluded that the only hope was a dogs-led relay. The serum would travel partway by train to Nenana, and from there, sled teams would haul it through nearly 700 miles of wilderness to Nome. Within days, more than 20 mushers volunteered. Each team would take a leg of the route, traveling by moonlight or under the northern lights, and hand off the vials like a relay baton. Conditions were merciless. Temperatures dropped below minus 60 degrees, paths vanished under blizzards, and frostbite ate at exposed skin. Dogs pushed forward through snowdrifts, and mushers clung to their sleds while ice lacerated their faces. The sealed glass vials of serum were latched down heavily. Even the knowledge that a fall could shatter them did not slow the riders. Everyone knew the freight weighed more than its ounces. It carried the lives of children waiting far ahead. Among the many mushers, two stood out. Leonard Zapala with his lead dog Togo endured the longest and most dangerous segment, more than 260 miles in just a few days, including a perilous crossing of Norton Sound across shifting sea ice. Winds tore across the frozen sheet and cracks threatened to swallow the team whole, but Togo led them steady until they reached land again. It was a gamble to gain nearly a day's time. Gunnar Cousin, driving the final team into Nome, faced a different peril. A whiteout storm erased all visibility, leaving him blind to the trail. His lead dog, Balto, relied on scent to guide the sled the last miles into town, bringing the serum safely to the waiting physician in the early hours of February 2nd. Physicians began administering doses immediately to children who were already showing signs of infection. Panic gave way to relief as cases stabilized rather than spiraling out of control. Thanks to the serum's delivery, the expected devastation never came. Out of Nome's roughly 1,400 residents, only 5 died during the outbreak, a staggering success considering what might have happened without the relay. Across America, headlines hailed it as the Great Race of Mercy. The dogs themselves became celebrated symbols. Statues of Balto, the final runner, still stand today, while many Alaskans argue that Togo's grueling journey across the ice was the true backbone of the rescue. Historians now credit Togo and Zapala with covering the hardest, most dangerous distance. Yet the point was never competition. Each musher and every dog formed a chain that held against the cold long enough for medicine to arrive where it was needed. The impact reached beyond immediate survival. The race reminded the nation how desperate communities remained when public health relied on emergency cures instead of prevention. Newspapers not only praised the heroism of the relay, but also asked the pressing question, what if serum had not been waiting in Anchorage? In the 1920s, diphtheria outbreaks in the United States numbered between 100,000 and 200,000 cases per year, with an estimated 13,000 to 15,000 deaths annually. The Nome race underscored that at any moment those numbers could climb higher if vaccine use remained patchy. Public health officials responded by pushing vaccination more forcefully across the country. By the mid-20th century, diphtheria cases in the United States fell sharply. Today, it is so rare that physicians may only encounter it in textbooks. Fewer than 5 cases have been recorded nationwide in the last decade. The memory of Nome became part of that trajectory. It showed in stark terms why inoculation was a safer path than gambling on the arrival of medicine through storms and darkness. Parents willingly brought children to clinics rather than risk the fate of their Alaskan counterparts. Yet the event lives on in cultural memory not only as a public health lesson, but also as a testament to endurance. The partnership of humans and animals became a story repeated in classrooms and communities, a moment when survival hinged not on technology, but on muscle, instinct, and refusal to quit. The Iditarod sled dog race, created decades later, traces its spirit to the serum run. Though now a sporting event, it still echoes the urgency and grit of the mushers who drove themselves and their dogs against impossible weather in 1925. Memory of the relay also reemerges whenever vaccine logistics face stress. In the early 2020s, as cold storage vaccines had to be delivered across continents during the pandemic, health officials drew parallels to Nome's experience a century earlier. The tools had changed. Cargo planes, thermal containers, and digital tracking replaced dogs and sleds, but the principle was unchanged. Public health collapses if medicine cannot cross the last mile in time. Almost a century later, the facts remain sharp. Thousands were saved because a chain of mushers and dogs carried fragile vials through frozen wilderness, and only a handful of lives were lost to diphtheria in Nome. The underlying lesson is just as sharp. Heroism can buy time, but prevention, through widespread vaccination and secure supply chains, brings lasting safety. The story of Nome is remembered for courage in the face of nature's cruelty, but it also warns that delay kills when communities rely on last-second rescues. That tension between protection and preventable harm appeared again in another chapter of American history, when the danger did not come from geography or weather, but from a policy decision that made ordinary citizens face lethal risk from the very government sworn to protect them. The government once poisoned alcohol. During Prohibition in the United States, a drink carried risk beyond arrest or fines. What many citizens never realized was that some of the alcohol reaching their glasses had been deliberately poisoned by their own government. This wasn't rumor or underworld sabotage. It was official policy. Federal authorities argued that if they could not stop bootleggers from diverting and redistilling industrial alcohol, they would make that industrial supply so unsafe that nobody would dare consume it. Manufacturers were ordered to add methanol, wood alcohol, to their stocks of industrial ethanol used in fuels, cleaning agents, and factory products. The idea was that anyone trying to repackage it as liquor would be met with deadly consequences. Methanol looked and smelled almost identical to drinkable ethanol, but the similarities ended there. Once ingested, methanol is metabolized into formaldehyde and formic acid in the body. These compounds ravage the central nervous system, cause irreparable damage to the optic nerves, and in many cases shut down organs entirely. Even small doses could result in permanent blindness. Larger amounts were fatal. Authorities knew methanol posed these dangers. That knowledge was precisely why they chose it as the deterrent. The practice of denaturing. Alcohol was not entirely new. Since 1906, industrial alcohol had been subject to additives to discourage drinking, a measure tied to tax enforcement rather than prohibition. But in 1926, under President Calvin Coolidge, the formulas were altered to increase methanol content sharply. It was a calculated escalation. Officials hoped that a more poisonous blend would collapse demand for bootleg liquor once the human cost became obvious. Bootleggers, however, did not back away. Their livelihoods depended on transforming industrial alcohol into something that resembled gin, whiskey, or rum. They were accustomed to crude distillation, cutting corners, and disguising impurities with flavorings. Removing methanol safely required precision chemistry they did not possess. The result was predictable. Industrial alcohol laced with methanol made its way into bottles across the country, sold in speakeasies and private gatherings under the guise of harmless liquor. For anyone pouring a glass, there was no way to know whether it would bring relaxation or immediate tragedy. Medical consequences soon arrived. Hospitals began seeing waves of patients after evenings of drinking. The pattern was unmistakable: splitting headaches, dizziness, nausea, followed by sudden blindness or collapse into seizures and coma. Doctors struggled to help, but effective treatments were scarce. Some survived, but lived the rest of their lives permanently blinded. Others never woke up at all. By the late 1920s, major cities recorded dozens of deaths in single weekends tied not to crime or violence, but to tainted alcohol. The cruelty of the policy rested in its unequal effects. Wealthier Americans often had access to safer, imported liquor smuggled past inspectors or purchased through private channels. Working-class neighborhoods had no such recourse. They consumed whatever local suppliers could provide, and those suppliers often relied on industrial alcohol. A deterrent meant to suppress nationwide drinking, instead imposed the severest punishment on communities with the fewest alternatives. Officials defended the approach on grounds of public health and deterrence. Their argument was stark. If people saw friends and neighbors sickened or blinded, they would reconsider drinking at all. But the reality was harsher. Instead of discouraging alcohol use, the policy eroded public faith in government. Ordinary families who wanted only to raise a glass at a wedding or holiday found themselves victims of a hazard introduced by federal mandate. Newspapers seized on the contradiction. Editorials called it a state-sponsored tragedy, a punishment wildly disproportionate to the offense. The outrage built with each new story. Newspapers publicized names of the dead, often framing them as victims of misguided policy rather than reckless drinkers. Ministers, physicians, and even some politicians who supported prohibition in principle condemned the poisoning practice. The phrase "chemist war prohibition" appeared in print, describing the campaign as an unintended moral catastrophe, an effort to save lives from alcohol abuse that instead destroyed lives with lethal chemistry. Estimates of the toll vary, but conservative figures suggest more than 10,000 people died from methanol-contaminated alcohol during Prohibition. Thousands more were left blinded or incapacitated for life. Even as evidence mounted, the federal government resisted acknowledging responsibility. Officials insisted that those who drank illegally assumed the risk. Behind closed doors, however, pressure grew. Physicians argued that enforcement could not justify deliberate poisoning. Lawmakers worried that public opinion might turn decisively against prohibition as a whole. While the official stance remained defensive, formulas for denaturing industrial alcohol were quietly altered again, reducing methanol content in later years. By the time Prohibition ended in 1933, the poisoning program was remembered as one of its darkest legacies. The 18th Amendment had promised sobriety and morality. Instead, it delivered organized crime, rampant black markets, and a government willing to sanction a policy that left thousands dead or disabled. Historians still debate whether officials underestimated the risk or knowingly accepted it as collateral damage. Either way, the outcome was the same. Lives were lost not because of disease or contamination, but because deterrence had been confused with protection. The aftermath shaped how regulators approached public health in the decades that followed. With Prohibition repealed, authorities shifted focus to ensuring that alcohol produced and sold legally was consistently safe. Inspections, licensing, and taxation replaced sabotage as tools of enforcement. A new philosophy took hold, that governments could penalize illegal acts through fines or restrictions, but must not endanger the basic safety of consumers. Protecting citizens, even those who made risky choices, became the boundary line that agencies no longer dared cross. That shift influenced more than alcohol regulation. Food and Drug Oversight also absorbed its lesson. The FDA, not yet trusted in its early years, gradually grew into a role where guarding safety was the first requirement of legitimacy. Consumers learned that bottles and packages sold through legal channels were meant to carry implicit guarantees. The federal government was no longer free to use chemistry as a weapon in moral campaigns. The line drawn after Prohibition is still visible today. Regulatory systems now strive for transparency and harm reduction instead of deterrence through danger. When a food product is contaminated or a drug batch proves unsafe, governments issue recalls, quarantine supplies, and inform the public. The immediate aim is not to frighten people into abstaining but to remove the source of hazard. Death or blindness is not considered acceptable collateral on the road to social policy. Mistakes remain both abroad and at home, but the idea that a government would intentionally spike daily products with poisons is hard to imagine now. That unthinkable policy was only a century ago, a reminder that even modern-looking systems can carry blind spots that trade safety for expedience. Prohibition's poisoning program showed that the difference between deterrence and disaster can be as small as a single additive. In retrospect, the poisoned alcohol strategy became a cautionary boundary for science and governance alike. It made clear that unintended consequences must count as outcomes in themselves, not as acceptable byproducts. Any policy that gambles with the safety of ordinary lives shapes not only its own era but also the frameworks that follow. The failures of those years cast a long shadow, one that still frames how experiments are designed and how risks are weighed. And they remind us that the path from reckless enforcement to careful safety checks has been neither smooth nor simple. Yet, out of those early mistakes, science and policy would eventually learn to replace direct danger with prediction, protection, and foresight. From chaos to AI labs, today's safer science. Just over 100 years ago, progress in science often meant exposing healthy volunteers to real poisons or rolling out untested medicines to unsuspecting patients. By 2025, progress looks very different. The most dangerous parts of early research, gauging what might harm or heal, are increasingly staged first inside computers. Before a drug enters human trials or a new food reaches the market, it is now more common that algorithms have already examined millions of possible scenarios, identifying where the risk might lie and which options deserve real-world testing. What once relied on ordeal or accident is now filtered through simulation. This new approach reflects a fundamental change in how science structures its work. Where early investigators waited for painful evidence before drawing conclusions, modern labs start with prediction. AI models draw on decades of toxicology records, chemical structures, and clinical results. When presented with a candidate compound, the system compares it against known families of molecules and generates probabilities. This one may stress the liver, that one may interfere with neurological signaling, another might be safely metabolized. 40 years ago, narrowing down possibilities required long, expensive sequences of animal trials. Today, preliminary results can appear within hours on a screen, reducing the reliance on large numbers of animals and avoiding unnecessary exposure of volunteers in early phases. The goal is not to bypass traditional validation, but to focus experiments where they matter most. The scale at which these models operate is difficult to fathom. A human researcher might spend weeks studying the behavior of a single protein interaction, but an AI system can scan tens of thousands of variants in a single day. Pharmaceutical companies now use this capacity to prune vast chemical libraries, cutting down potentially billions of molecular designs to the few hundred worth pursuing. Food technology firms do much the same with proteins designed to mimic or replace animal products. This efficiency saves money, but more importantly, it reduces the number of unsafe candidates reaching either animals or people. Safety, once established slowly through observation of harm, is now anticipated earlier. Lab-grown meat illustrates how this predictive shift reaches beyond pharmaceuticals. In these projects, animal cells are grown into edible tissues without traditional farming. A generation ago, regulators would have taken decades to assess whether such products were safe to eat, requiring extended feeding studies and limited pilot markets. In 2025, early computational screens compare these cultured proteins against databases of allergens and immune responses. Simulations flag any structural similarities likely to trigger reactions before tastings ever begin. This does not erase the need for trials, but it removes much of the uncertainty long before a product reaches a consumer's plate. Drug development pushes the technique even further. Researchers use AI to predict which chemical scaffolds could bind to viral proteins or suppress tumor growth. Systems trained on genetic data now model how viral strains may mutate, offering vaccine designers a head start. During the early 2020s, an mRNA vaccine could take months to design against a new virus. Today, candidate formulations often appear within days of posting a genome sequence to global databases. Experts still rely on laboratory testing to prove these predictions, but AI compresses months of target discovery into hours, saving resources and accelerating critical timelines in crises. Generative AI has even been used to explore wider chemical spaces than humans could consider, suggesting molecules with novel shapes and properties. But even here, companies rely on lab-in-the-loop methods. AI generates possibilities, and then wet lab scientists confirm or discard them. Toxicology itself has also grown more nuanced under computational approaches. Instead of focusing only on immediate signs of poisoning, new models anticipate long-term ecological and biological risks. Algorithms can simulate whether pesticides may disrupt hormones, accumulate in tissues, or persist in the environment for decades. Farmers using new compounds can be warned about risks years before a real contamination event would have revealed them. Environmental reviews, once reactive to disaster, can now be proactive, sketching likely hazard in advance. Such foresight does not eliminate risk, but it shifts the battlefield. Damage is intercepted in data rather than after the fact in communities. Yet the results are not infallible. Every AI model depends on past data, and when new compounds stray far from known categories, predictions weaken. Scientists worry that speed could create blind spots, discoveries rushed into trials without sufficient cross-checking. One emerging concern is AI hallucination, where systems generate plausible but misleading answers that do not align with real data. Experts caution that transparency in algorithms, systematic validation, and continuous human oversight must remain central. Most observers agree AI is not a replacement for laboratories or clinical trials. It is a powerful filter that demands careful interpretation. Regulatory frameworks have begun adjusting to this reality. In 2025, guidelines such as the ICH E6, R3, modernization emphasized data governance and requirements for decentralized and digital trials. The FDA and other bodies issued new directions on how results from AI-driven models should feed into conventional pipelines, clarifying where simulation can accelerate steps and where physical validation remains non-negotiable. These moves reveal how oversight now revolves not just around chemistry, but also around algorithms, ensuring that the promise of acceleration does not erode the principles established after past tragedies. For working scientists, this means the role has shifted from enduring risk to managing uncertainty. A century ago, fate rested on whether volunteers collapsed after swallowing an additive. Today, progress means interpreting endless heat maps, probability curves, and risk bands, deciding which deserve experimental confirmation. This may seem less dramatic, but it represents a culture of research that aims to anticipate harm before it happens. The excitement of discovery has been joined by the responsibility of interrogation, knowing when software suggestions hold true and when they may mislead. The transition is not without its struggles. Competition among startups, investors, and nations encourages speed. Announcements of breakthrough drugs sometimes follow days-old simulations rather than years of experiment, raising concerns about hype outpacing substance. Meanwhile, regulatory bodies try to ensure that safeguards once applied in test kitchens and animal houses are not forgotten in server farms and data streams. In practice, this means that even as AI handles early screening, the later pillars of science remain in place: careful animal studies, controlled human trials, phased approvals, and post-market surveillance. Despite these caveats, the shift is profound. Where past generations tested safety by chance or disaster, today's researchers lean first on predictive tools that spare many from exposure. Consumers in 2025 may not notice the models running in the background, but the medicines filling pharmacy shelves and the foods entering restaurants already carry the fingerprint of algorithmic screening. Governments now ask companies not only to prove that their products are safe, but also to show how digital tools assisted in preparing that proof. The result is an environment both faster and more cautious. Fast because predictions accelerate discovery, cautious because regulators insist those predictions be confirmed in traditional stages. Scientists continue to balance ambition with restraint, innovation with oversight. They build on the lessons encoded by past mistakes without relying on sacrifice to move forward. And as the landscape keeps changing, with new risks surfacing in molecules, microbes, and data itself, the challenge is to keep tools and ethics advancing together. This moment, the pressure to trust prediction while demanding proof, captures the core tension of modern research. It's a tension that would have been unrecognizable to those who faced peril directly in the lab a century ago. And yet, much like them, we find ourselves asking what level of risk is justified in the name of progress, and who should bear it. That question continues to shape science in every era, and it will guide whatever judgments the future makes about ours. The reckless trials of the past, volunteers swallowing preservatives, families harmed by untested drugs, even dog teams racing through storms, forced medicine and science to build the safety frameworks we now depend on. Those moments of risk and tragedy set the stage for today's more cautious systems, where AI filters thousands of candidates before they ever reach people or animals. Still, prediction is only one layer. Ethics, oversight, and regulation ultimately decide whether progress is safe. History suggests we'll be judged by how well we balance innovation with safeguards, a debate echoed in today's policy updates. Which story surprised you most, and would you choose speed or caution?